Hello and welcome to Nikolay's genetics lessons, and today's problem is: a scientist performs a series of experiments to determine the recombination frequencies between the following genes. He acquires the following data: WX 3% recombination frequency, XY 2%, YZ 13%, ZW 8%. Which of the following choices place the genes in the correct order relative to one another? And here's the variants to choose from. Everyone can solve this type of problems. The only problem here is the most efficient way how we can do it. And this is exactly what I want to show you today. First of all, let's find two genes with the greatest distance between them. And this is going to be fragment Y and Z with 13% recombination frequency. That means that between Y and Z we have greatest distance if we compare with other fragments. Let's plot this fragment here. So let's say this is going to be our fragment, and we have gene Y here and gene Z here, and in between them we have recombination frequency which is 13%, which directly correlates to the map units, 13 map units, or we can also say 13 centimorgans. Now let's find second fragment which is greatest, and this is going to be ZW with 8% recombination frequency. So we have Z here, so we have ZW. That means that we can put W with here with 8% recombination frequency between Z and W, or we also can put gene W, say here, with also the same 8% recombination frequency. So we have now two hypotheses, whether gene W is going to be here or it's going to be here. By the way, this fragment here, take a look, we have 13% and 8% here, so 13 minus 8 is going to be 5. So this fragment here is 5% recombination frequency. So if gene W and Y separated by this distance, we expect between these two genes 5% recombination frequency. Now let's take a third fragment in length. It is W and X. So we can put X, for example, here with 3% recombination frequency, or here, or here, or the last variant would be to put it somewhere here. So WX, so we put X here, 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 and here. Now let's take a look. Between gene X and Y, recombination frequency is 2%. So let me underline. Now let's ask a question. Can 2% recombination frequency be between gene X and Y? No way. No way. So we can exclude this position. Can be recombination frequency between X and Y be 2% here? No way. We exclude this position for gene X here also. Can be 2% recombination frequency between gene Y and X here? No way, because this fragment is 5%. So we can exclude this position of the gene X here. So this is the only correct position for the gene X here, and we are going to have 3% recombination frequency here and 2% recombination frequency here, and we know that this fragment is 5% recombination frequency. And we can exclude now this fragment also, and here's the correct sequence. Gene Y, so let me put gene Y here, then gene X, then gene W, then gene Z. So this is our sequence. Let's check. Do we have such sequence here? And actually none of the sequences start with gene Y. But you know what? The sequence is the same as following sequence. So Z, W, X, and Y. Because we can flip the sequence, and this is going to be the same sequence but in different order. And we only can know the order, but we cannot say that this is going to be first gene and this is going to be the last gene. This also can be the first gene and this is the last, but sequence is going to be the same. Now let's check the sequence among the sequences here. And as you see, this is answer C. And this is all for today. Subscribe and see you in the next video.